Thank you. So, optimized FDR estimation using p-value scoring. This work has been done in collaboration with David Fenyö of the New York University. So, FDR means false discovery rate, and here it's FDR in the context of peptide identification in proteomics experiments. So just a reminder of the workflow of a proteomics experiment. So a protein mixture is taken from some organism and is separated and digested into proteolytic peptides, which are typically separated before they are subjected to mass spectrometry. So typically an individual proteolytic peptide is isolated and fragmented, and the resulting information is a fragment mass spectrum, which is then used to compare with information from the sequence collection, and supposedly it will yield an identification. And that is possible because with the theoretical sequences, you can do the same thing as in the experiment. You can digest with the same specificity as you had in the experiment enzyme. You can compute the masses of each peptide. You can fragment each peptide theoretically according to the same rules as is applied in the mass spectrometer. And you end up with a theoretical mass spectrum for each peptide in the sequence collection, which you can compare with your experiment. And in each comparison, there is some kind of computation that yields a score. So if you have a set of experiments, it means you will have a distribution of scores for the highest ranked peptide in each experiment, in each comparison. However, identification results can be false, and false identification is caused by random matching. And in this picture, we see to the left, we see the average number of matching peptides as a function of the proteolytic peptide mass. And the three different panels represent three different proteomes. So the larger the proteome, the larger the number of randomly matching peptides. And the top panel represents the human proteome, and the different lines you see there in different colors represent different mass accuracies, going from the top at 10 ppm and down to 0.1 ppm. So if we look at the mass of 2,000 Daltons and the very good accuracy of 1 ppm, in the human proteome we still have, on the average, 4 to 5 peptides. We also notice that as we go up in mass, the number of matching peptides is going down. So since we have this random matching, we need some criterion for testing the statistical significance of an identification result. And that we can do once the distribution of scores for false results is known. And this could be representing such a distribution of scores for false results. So if we do an experiment which yields a particular score, we can look at the overlap with that distribution, and this red area is then representing the significance level or the p-value. So the further away our score is from this distribution, the lower is the p-value. The true results have typically unknown distributions. Just we can hope that they are far away from the false results, but in reality they often overlap. So the distribution of scores for false results can be determined by different methods. One can collect statistics during each search. That's a method which is used in the X-tandem algorithm, and I will not talk about that today. One can use direct calculation, as we do in the Probity algorithm, which I will talk about later. Or you can use computer simulation. And computer simulation can be done by doing repeated searching using fake data and real sequences, or realistic data and fake sequences, or decoy sequences. So the realistic data and fake sequences are often used to estimate the false discovery rate for a set of identification results. And the false discovery rate is particularly interesting in large data sets with many identifications, since you have a multiple testing. So the FDR would then be defined as the number of results when you search the fake sequences that are better than a particular score, divided by the number of results when you search the real sequences that are better than this particular score. This can also be envisioned as the ratio between this red area and dashed area when you look at the distributions of scores when you search the fake sequences and the real sequences, respectively. The score distributions for false results may also move around. So they depend on the search conditions. So for instance, they depend on the size of the sequence collection that you're searching. They depend on the mass accuracy, depend on the assumption about the number of missed cleavages. In a set of spectra in an experiment, you typically run with the same search conditions. So you search the same sequence collection with the same boundary conditions. Means that this is not necessarily an issue. However, the data also have different features. So different spectra contain different number of fragment peaks, and they also have different precursor mass values. So if we take a look at false identification results using fake data and real sequences, and this is the distribution of scores when using the number of matches as the score. This is the cumulative relative frequency, and these three different distributions represent spectra containing 5 fragments, 15 fragments, and 50 fragments. So we see that these definitely yield different score distributions for false results. However, if we use an algorithm that is using p-value scoring, we see that these 3 different cases, they collapse onto the same line, which is diagonal here, and it's the frequency here as a function of the score. The p-value is the power 1, which means that this is an observation from a uniform distribution, as it should be if we're dealing with a p-value. So the algorithm that has produced this is the Probity algorithm, which was originally designed for peptide mass fingerprinting, now converted to deal with ASMS data. So it basically takes into account all the features of a spectrum, and also models the particular peptide you're comparing it with. And the conversion into a p-value, which is called S in this graph, is actually done by taking into account the number of sequences that are participating in each search. So moving back to scoring with the number of matches and looking at the effect of the precursor mass of the data. So this is a simulation comparing 1,500 Dalton peptides and 2,500 Dalton peptides. And we also see that these distributions separate distinctly. It doesn't matter whether we use real data and fake sequences or fake data and real sequences. So this separation between 1,500 and 2,500 is the same. So now, if we construct spectra that contain 15 fragment masses, but with poor quality, so they are 80% background, but they are still real, representing peptides, and we search real sequences, we can get a distribution of scores for those two mass values. And since we have now both a distribution for searching the real sequences and a distribution for searching the fake sequences, we can do an FDR computation for this case. And if we look at the red and the black curves here, we see that is what the FDR would be as a function of the number of matches if we were treating 1,500 separately and 2,500 separately. However, if you're not aware of this effect, you may lump all your data together, and that is what is assigned both here. We also notice that the blue line representing both is closer to 1,500 than it is to 2,500, and that is because the underlying data in this case are representing the abundance in nature of these two mass values. So 1,500 is more common than 2,500. Anyway, the result here is that if we have lumped everything together and we try to choose an FDR and put a number of matches threshold, we will somewhat underestimate the false discovery rate for 1,500 Dalton, and we will throw away 2,500 Dalton results unnecessarily. So if we then instead use the p-value scoring, these false result distributions, then they again then collapse into this line, which is coming from a uniform distribution, and it doesn't matter again if we are using fake data or if we are using fake sequences, and it doesn't matter if we have 1,500 or 2,500. It's all following a uniform distribution. And that allows us to use that uniform distribution for the FDR estimation. So basically we say that if we use that, we don't need the decoy searching. That is obsolete. So we just compute the FDR as the ratio between the p-value and the relative cumulative frequency when we search the real sequences, that the relative cumulative frequencies up to the p-value. And this graph shows it's for the same simulation as previously, but using the p-value scoring. So this is the 80% background data, and the green line is the distribution of p-values for all the results, and the blue line is the uniform distribution, and the red dots is the FDR computed in this fashion. So, in conclusion, the p-value scoring leads to an unbiased computation of FDR, and the decoy searching becomes obsolete. So I would like to acknowledge the collaboration with David Fenyö, the funding from the Swedish Research Council and the Carl Trygge Foundation. I would like to thank the organizers for the opportunity to present this work, and I would like to thank you for your attention.
So we have 2 minutes for questions.
Hi, I have 2 questions. First, you commented that your p-values are accurate, rigorous, which is very good. But there is another interest that if somebody uses your p-value, then can the person increase the number of identifications at the same FDR, like Percolator does or other programs does? That is the first question. And my second question is whether is your method can your method be generalized to like more complex scoring functions? Because you showed that it works for a very simple scoring function for counting number of matched fragments, but that is not the scoring function that are used in the field anymore. So,
yeah. So if I take the last question first, so, I took the number of matches because it's very straightforward to look at the results of it. But basically, any scoring that doesn't yield the same score distribution at any condition will give you this error in the FDR computation. So then you basically have to look at your scoring function and see how it performs. And the first question, whether it will be better, well, in these examples I showed you, if we use the number of matches as the scoring, it's so poorly discerning the random distribution from good results, so that it's quite obvious for that reason that you will get, when you do the FDR analysis using the p-value instead, you will gain.
Thank you very much. So in the interest of time, we need to go on as well, but please stop by at the end of the session if you have more questions.